SUMO binding entities make it possible to isolate endogenous SUMO-related proteins in vivo. This is rather challenging due to the presence of active SUMO-specific protease and the low fractions of SUMO-related proteins in vivo.
The isolation of the SUMO-related proteome by using SUMO binding entities is advantageous as it offers an increase in the overall affinity for SUMO substrates. This is due to the fact that SUBs are recombinant proteins that comprise tandem repeats of SIMs, thereby recognizing specifically SUMO molecules on modified proteins.
The use of SUMO binding entities for the isolation and characterization of the SUMO-related proteome in liver cancer is a fast and sensitive method. It provides vast information on the rather unknown role of the SUMO-elation pathway in liver cancer and a potential therapeutic approach.
SUMO binding entities can be used to isolate and characterize the SUMO-related proteome in other tissues besides the liver, both from human specimens and animal models, as well as in several cellular models.
Although this technique is easy to perform, care should be taken when analyzing several samples at the same time, as confusion could occur due to the several steps involved. Therefore, we recommend careful labeling of all the tubes before initiating the experiment.
The visual demonstration of the use of SUMO binding entities facilitates the reproduction of this protocol, especially due to the difficulties of handling the beads and the eventual loss of material during the protocol.
Human hepatoma cells have been previously prepared by plating cells in P100 plates at a density of 1.2 to 1.5 million cells per dish and maintain them in standard growth media at 37 degrees Celsius. When ready to use the cells, aspirate the media from the plates and wash them with 5 milliliters of sterile PBS. Put the plate on ice and add 500 microliters of lysis buffer supplemented according to the manuscript directions. Then, use a cell scraper to gently scrape the cells off the bottom of the plate into the lysis medium. Alternatively, harvest the cells by trypsinization. And add 1 milliliter of trypsin EDTA to the plate, making sure that the cells are covered. Put the plate in a 37 degrees Celsius incubator for 5 minutes to allow them to detach from the plate, then add 2 milliliters of pre-warmed growth medium to stop the trypsinization. Centrifuge the cells at 150 times G for 10 minutes and aspirate the supernatant. Then wash the cells with PBS and centrifuge for another 10 minutes. Aspirate the supernatant and add 500 microliters of lysis buffer. Centrifuge the lysed cells at 15,000 times G and 4 degrees Celsius for 10 minutes, then transfer the supernatant to a fresh tube and discard the pellet. When ready to use the harvested mouse livers, homogenize 75 mg of fresh or snap-frozen fragments in 1 milliliter of ice-cold lysis buffer. Run the homogenizer according to the manuscript directions. After tissue homogenization, centrifuge the samples at 15,000 times G and 4 degrees Celsius for 10 minutes, transfer the supernatant to a fresh tube, and discard the pellet. Prepare glutathione beads by adding 1 milliliter of deionized water to 70 milligrams of beads and reconstituting them overnight at 4 degrees Celsius. After swelling, wash the beads 3 times with 10 milliliters of deionized water or PBS, centrifuging at 300 times G for 5 minutes after each wash. After the washes, resuspend the beads in 1 milliliter of PBS to obtain a 50% slurry. For each sample, add 100 micrograms of GST subs or GST control to 100 microliters of the glutathione bead slurry and 500 microliters of PBS. Incubate the mixture at 4 degrees Celsius for at least 2 hours while rotating, then recover the beads by centrifugation at 300 times G for 5 minutes, and resuspend them in PBS. Take 10% of the previously prepared cell lysate and dilute it in an equal volume of 3x boiling buffer. Add 450 microliters of the clarified lysate to 100 microliters of the glutathione bead slurry. Incubate the lysate with the beads at 4 degrees Celsius for at least 2 hours while slowly rotating. After the incubation, spin down the beads at 300 times G for 5 minutes and collect the supernatant for analysis. Transfer 10% of the total volume to a separate tube and add an equal volume of 3x boiling buffer. Wash the remaining sample 3 times by adding 1 milliliter of ice-cold PBS with 0.05% tween 20 and spinning it down at 300 times G and 4 degrees Celsius for 1 minute. Carefully aspirate the supernatant, making sure that no liquid remains. Then, elute the sample with 15 microliters of 3x boiling buffer and 15 microliters of lysis buffer. Run a Western blot analysis according to the manuscript directions. If performing mass spectrometry, desalt the peptides using stage tip C18 microcolumns and resuspend them in 0.1% formic acid prior to analysis. Load the samples onto an LC-MS system and analyze them in triplicate. Proceed with protein identification and abundance calculation using the associated software. Perform statistical analysis according to the manuscript directions. This protocol has been used to isolate SUMO-elated proteins in mice with glycine N-methyltransferase deficiency, which causes spontaneous development of liver cancer and their wild-type littermates. Ponceau S staining was performed on the input, flow-through, and bound fractions from the SUBS pull-down assay. Western blot analysis of LKB1 captured with SUBS shows that the levels of LKB1 SUMO-elation are augmented in liver tumors. Human hepatoma cells and non-transformed liver epithelial cells were used to investigate the capacity of the SUMO trap to interact with naturally SUMO-elated proteins. First, conventional protein staining was used to visualize the total material captured with SUBS. Then, mass spectrometry showed that 742 proteins were enriched in the hepatoma cell SUB sample, and 577 were enriched in the liver epithelial non-transformed cell SUB sample.
When performing experiments with SUBs, it is important to maintain cells and tissues on ice and add specific inhibitors to the lysis buffer in order to maintain the proteome SUMO-elated.
After the isolation of the SUMO-elated proteome with SUBs, we can further characterize it by using either Western blot analysis or mass spectrometry.
The application of the SUBs to isolate and characterize the SUMO-elated proteome in liver cancer tissues and hepatoma cells is paving the way to explore the role of the post-translational modifications of SUMO in liver cancer, which may provide a potential therapeutic mechanism.